Thank you for the invitation to come here today and talk about my work. So I'm going to talk about, the title of my, talk is Soil Health, Opening the Black Box in Specialty Crop Production Systems. And today I'm going to first just briefly introduce soil health and the relevance of soil and plant microbiomes to soil health. Then I'm going to talk about disease-suppressive soil and biological control of pathogens, which is an area of soil health and relationships between soil and plant health that we're very interested in my lab. Then I'll go through several examples of research projects that we're doing to try to connect these two factors, and then we'll have some time for questions. So first of all, there's a lot of interest these days in soil health, what it means, why farmers should be thinking about trying to manage healthy soils on their farms. And while there's a lot of information out there demonstrating how soil health is important in providing various ecosystem services, what farmers really want to know is how does it exactly help the health of their crops. And so that's something that we've been very focused in, again, at my lab, is trying to understand these relationships. So how can we manage soil health specifically to improve the health and productivity of crops? So before we start talking about that, it's important to just refresh ourselves on exactly what soil health is. And so these kind of Venn diagrams are often used to describe what healthy soil is. And it's this combination of these various properties. And so things like chemical properties, soil pH, cation exchange capacity. Farmers are pretty well versed in this and knowing that these are very important in providing the fertility for their crops. And so they often send soil tests off for these types of soil properties and are managing them. So another important factor of soil health is the physical properties, so things like bulk density and aggregate stability, which are really important for water infiltration and how roots and things can move in soil. And this is also pretty well understood. There are some soil tests out there and testing labs that can do this, but they're not as often as applied. Then lastly, we have the biological component, which is also very critical, but it tends to be the least understood and implied, but could actually be the most important. And so that's today what I'm going to focus on, and specifically a component of soil biology, and that's this idea of these soil and plant microbiomes, which has always been looked at as kind of a black box because they've been so hard to study. But within the last 10 years, we've had the development of new molecular tools that are allowing us to explore these soil microbiomes in much greater depth. And so that's what we study in my lab. I'm a soil microbiologist, and we're specifically interested in this tripart relationship between soils, plants, and the beneficial microbes that live in soils. And how can we learn more about these communities and find ways to leverage them to enhance the health of plants. And so that brings us to plant microbiomes, which is something that has been getting a lot of press recently. So we know, just like soils, plants are literally covered inside and out with microorganisms. So they're living on the surface of leaves, they're living on the surface of roots, and they're even living inside the plant. And so most of these communities that inhabit plants come from the soil. And while it may seem hard to believe, just like us, plants actually are more microbes than they are plants because the number of microbial cells actually outnumbers those of the plants. And so there's a lot of interest in understanding what exactly are these microbes doing. And so there's a lot that we do know and a lot that we don't know. But what we do know is that some of these microbes can be bad. So things like plant pathogens, we've known for a long time, and these have been very well studied, that these microbes can negatively impact the health of plants. We now know that there's some foodborne pathogens, things like Salmonella and E. coli, that are really bad. So these don't necessarily negatively affect plants, but they can attach to them and negatively affect our health when we eat these plants. But there's a whole lot of other microbes that are interacting with plants, either through the soil or the plant microbiome, that aid in plant health. So they're altering plant physiological properties, helping plants get nutrients, and interacting with these pathogens to suppress them, so keeping the plant and foodborne pathogens off of plants. And so consequently, learning more about the composition of these microbiomes is a key determinant of the health and productivity of plants. They're very important. And so some have proposed that this is what the future of agriculture is going to look like. Just like we can go out and take a soil test for soil chemical properties and understand that, oh, our pH is low or we need these nutrients and we can add these. Someday we might get to the point where we can actually do a soil microbiome report and know which microbes are deficient in our soils and need to be added. But we're still a long way from this point. So while there have been some good microbes that have been isolated and are now being developed for use as inoculants, which we'll talk more about later, the survival and efficacy in the field is often variable for reasons that we're still trying to figure out. Then the rest, the identity and awareness of many of these other good microbes in soil, is still unclear. And so again, that's what we're really trying to figure out in my lab, is how can we learn more about these microbes, how to manage them, how to shift these communities in ways that can help farmers increase the productivity of their crops while also needing to rely hopefully less on off-farm inputs and pesticides to keep their crops healthy. And so that brings us to one of the things that we're very interested in my lab, and that's how to manage pathogens that cause disease. So most farmers are probably aware, they've seen symptoms of this in the field, that there are a lot of bad microbes that can attach to plants and really negatively impact the quality of fruit or destroy the entire crop. And so while there is strong varietal resistance to some of these pathogens, other pathogens there's not. And then while there are pesticides that are available in many cases to control these crops, some of them can have negative impacts on other organisms, or maybe we don't have the right pesticide to control some of these pathogens. Lastly, we know that pathogens themselves are constantly evolving. Even when we select varieties that have resistance, the pathogen is going to evolve to try to overcome that resistance. And they're also becoming resistant to some of the pesticides that we use. Consequently, we need alternative control strategies. And so that brings us to a topic that I'm really interested in, and that's disease suppressive soil. And so the idea of that is basically, when you look at this little picture down below on the right, is a conducive soil. And this is what we would expect. There's the presence of a problematic pathogen, the environmental conditions are good for that pathogen, and you have a susceptible host, and so the plants are negatively affected. But we also know there's these so-called disease suppressive soils where, despite all these factors, the plants remain really healthy and viable. And this was first documented in France in the 1880s with Fusarium wilt and cotton and pea, but it's since been observed in multiple systems worldwide, so we know it's not an isolated phenomenon. And there's been quite a bit of work to demonstrate really that this is due to biological processes. So if you take those suppressive soils and pasteurize them to kill the organisms living in them, then they act more like a conducive soil. And this is due to a number of different direct and indirect mechanisms. So these are some of the direct mechanisms of pathogen suppression in these disease suppressive soils. So there can be competition, where all these beneficial microbes living in these soils actually outcompete the pathogens for space and nutrients, like you see in the kind of top left corner there diagram with all those microbes living on a root. There can be antibiosis, where this beneficial microbe is producing compounds like antibiotics that we commonly take. That's where our antibiotics come from. It's from soil microbes, but they can also directly kill pathogens, like you see in the top right-hand corner, where that strip of bacteria on the left is slowly killing that furry fungi growing in that plate. So you can also have predation and parasitism, where the beneficial microbe actually preys on and destroys a pathogen, like in the bottom left-hand corner, where there's a good fungal organism kind of coiling around a bad one and piercing it and negatively affecting it. And lastly, signal interference, where the beneficial microbe just interferes with the signaling processes of the pathogen in the plant. So next, there are indirect mechanisms of pathogen suppression. And so this might be the case where a microbes in the soil are enhancing the health of the plant by helping them acquire nutrients and water, and so the plant's just more healthy and able to withstand pathogen pressure. Then there's also this phenomenon that we're very interested in my lab called induced systemic resistance, where the beneficial microbes actually elicit an immune response in the plant. So it's basically like the plant stimulates and upregulates its immune system so that when it is attacked later, it's able to respond much more quickly to shut down the pathogen, similar to like a vaccine that we would think about for ourselves and withstanding attack by viruses. So we're interested in this phenomenon, and we're trying to look at kind of 3 different ways of how we can support more biological control of pathogens so that we can ensure these systems and this way of managing pathogens is helpful for growers. And so one of those we're going to talk about first is improving soil health. So thinking about how we can stimulate those beneficial microbes in soil. Second, we're going to talk about plant breeding and some of the things that we're doing to interact with plant breeders in selecting for microbes, beneficial microbes. And then lastly, how can we improve the efficacy of these beneficial microbial inoculants? So first, again, we're going to talk about promoting biological control of pathogens by just improving soil health. And there's many ways potentially to do this in soil, and so in my lab we're looking at various ways to stimulate soil health. But today I'm just going to give you a couple examples on using cover crops to promote these beneficial microbes. And so probably, again, cover crops are something that's been getting a lot of attention and a lot of farmers are adopting it, and that's great. And so we know, a cover crop just by definition is a non-cash crop grown for the protection and enrichment of soil. And it tends to be the most cost-effective way to improve the health of the soil. So how it's improving soil health and potentially for disease-suppressive soils is that these crops are creating pores for greater aeration and storage of water in the soil. That's really important to support soil microbial communities. But probably even more importantly, the cover crop is providing carbon compounds to the microbes, both through the root exudates, the carbon compounds, the photosynthates that the cover crop is generating. Some of those are leaking out of the root system, and then it's the residue of those cover crops that go back into the soil. And the reason this is important is if you look at that little figure in the top right corner of the slide, when we think about plants, they are generally nutrient limited, right? So we're thinking about adding nitrogen and things like that because they get carbon from the atmosphere and fix that through photosynthesis. But microbes below ground are actually carbon limited, so they have access to nutrients, but they need carbon. And so keeping new inputs of carbon into the soil is really important to support these beneficial microbes. And so as a couple of examples of that, I'm going to give you 2 examples of research projects that we've done. And so one of those is a study that we did a little while ago, but we've followed it up with some additional studies that I think you'll find interesting. And so one of those, this first one, is looking at leguminous cover crops in high tunnel production systems. So high tunnels are another thing that's just increasing rapidly here in the Midwest as people are interested in local food systems and supplying more specialty crops to local markets. And high tunnels have numerous benefits. So they can extend the growing season, they can protect plants from some pests, and they can increase yield. But they also have some drawbacks, and one of those, most importantly, is soil quality degradation. So these are very intensively managed systems. You know, it's expensive real estate for farmers, and so they're growing high-value cash crops like tomato over and over again and not thinking so critically about soil health in these systems. And so potentially you can degrade the soil over the time and build up soil-borne pathogens in these systems. And so these are the results that we found in those trials. And so if you look at the top two figures, that's pepper yield. And then on the x-axis, you can see the different treatments. So again, the control, the conventional, the green manure, and the chicken manure. And we have the open field on the left and the high tunnel on the right. And so as expected, the high tunnels increased pepper yield. You have warmer conditions, longer season, and luckily, of course, the control was deficient in fertility, as we knew, but all three of the fertilizer treatments that we evaluated were able to meet pepper needs in both of these systems, even in the high tunnel system where we have much greater productivity. So what was also interesting is that we saw some really interesting trends and changes in soil health over time. And so if you look at the bottom two figures, this is active soil carbon. And we have the same 4 treatments, but they're in blocks over time. So for example, on the bottom one on the left, we have 2011, 2012, 2013. And so what we saw is, soil quality certainly declined in the high tunnel conventional treatment. So this was just inorganic fertilizers. These are kind of the yellow bars. You can see a decrease over time in the figure on the right as the soils were being depleted and the plants were really scavenging nutrients from those systems. But we saw that the green manure especially increased. It was able to not only sustain soil quality in these very intensively managed systems, but it was actually increasing the health of the soil over time. And so next we wanted to know, well, how could this affect pathogen dynamics? So we were lucky, I guess, and didn't see a lot of pathogens out in the field. And so what we decided to do then was to take these soils, we brought some into the greenhouse, and we specifically inoculated them with a soil-borne pathogen to see how resilient they would be in case a pathogen was to come into these systems. And so for these trials, we chose Rhizoctonia solani, which is a very ubiquitous soil-borne pathogen that affects a number of different crops. And for these assays, we chose a susceptible snap bean variety. It's very easy to grow in the greenhouse and really clearly demonstrates resistance or susceptibility to this pathogen. And so in those little pictures there, you can see the left-hand side is a snap bean plant that was very negatively affected by the Rhizoctonia, whereas the one on the right was not. You see a very nice healthy root system and healthy plants. So the results of that were kind of similar to some of the trends that we saw in the field. So first of all, in this little figure on the lower right-hand side, you can see that the treatments, again, are the 4 bars. On the left is the open field, and on the right is the high tunnel. So as we suspected in the high tunnel, we generally had lower, or I guess the soils were more susceptible to the Rhizoctonia pathogen. So these soils are being very intensively managed in the high tunnel, and so they're becoming more stressed. As you can see, for example, the red bar is that conventional, where we saw the soil quality going down. which is very troublesome. However, in the bar to the right of that kind of green color, that's the green manure treatment, where the plants did very well despite the presence of this Rhizoctonia solani pathogen. They were very resistant to it and were able to fight that off. And so we wanted to know more what was happening with the microbial community. So had we potentially shifted the microbial communities in these two treatments, and was that playing a role in this biological control in the case of green manure, or susceptibility in the case of the urea treatment? And so we collected the DNA, and we did some sequencing. And so this is the results of some of those studies. So this is, in this study, one of my former PhD students, Sahar Abdelrazek, was very interested in studying carrot endophyte communities. And so these are microbes that actually live inside the plant for at least part of their life cycle. And they're very interesting, but they've actually been understudied just because they're very difficult to isolate and they really don't show symptoms in plants. So we haven't really understood that they were even there for a very long time until we were able to develop these new molecular tools to study them. And so they're interesting because we know that for them to survive inside a plant, the plant must relax its immune system to let them in and let them survive so that they're not acting like a pathogen. So while they've been understudied, we're not sure what they do. It's likely that they probably have some beneficial roles. And so again, we wanted to know, and Sahar wanted to know, how do these changes that we saw in soil health in the field influence what was colonizing these carrot roots, and does that have a role in suppressing Alternaria dowsii? And so she looked at this in a number of different carrot genotypes. You'll see there on the right, and we'll talk more about that later and why all these different genotypes were important. So this is what she found. First of all, you can see on the kind of two bottom pictures on the left, those are just bacterial isolates that she was able to isolate out of the carrot taproots and fungal communities. Or these are individual isolates, so pretty different colors, different organisms. It was really interesting to see just what lives in a carrot root. And so she found, first of all, that these endophytes were more abundant, and they were also more diverse in carrot taproots grown in the organic system. So next, she took these isolates and actually tested their suppressive activity against the pathogen. So in the kind of top right of the slide, you can see in the middle is the Alternaria dowsii pathogen growing. It's black. And then on either side, you can see Petri dishes where she's streaked the isolates. In this case, they're both bacteria that she collected, and looked at how they would stop the spread of this pathogen. And so she found there were several isolates that could suppress this pathogen. And as you see in the figure, the blue bars are ones she found from the organic system. The orange are from the conventional system. And so there were a lot more in the organic system that had this suppressive activity against the pathogen. So then she took those and did some greenhouse studies and actually inoculated carrot plants with the pathogen and confirmed that, yes, these microbes could help the plant to survive this pathogen. So next, we're going to move into kind of a second aspect of what we study in my lab. So I mentioned a lot of our work is really focusing on the soil. How do we build these healthy populations in soil so that they fight pathogens? Farmers have to apply less pesticides and they have healthier plants. But another big important component we need to think about is the plant itself. And so that's why we're going to move into talking about our work in plant breeding. So plant breeding is something that, has been happening for a long time. So both humans, farmers traditionally, and now plant breeders have been selecting plants for at least the last 10,000 years. And so we know this is very valuable. So these improved varieties have greater yield, greater nutritional quality, And it's also been very successful in selecting for resistance to harmful pathogens, right? We know some genotypes have mechanisms that allow them to shut these pathogens down, at least for a time. We know that plants can evolve, or microbes can, the pathogens can evolve to overcome that resistance. But it's very successful, and so that continues to be a very important component of plant breeding programs. But what about alternatively? So we know that plants, there's genetic variability within plants' crops in reducing, keeping pathogens out. Shouldn't there also be the same thing for plants to select or interact with beneficial microbes? So if we know plants can keep them out, maybe they can do specific things to keep them in and allow them or promote their activity in soil or let them come in, as in the case of endophytes. And so now I'm going to give 2 examples of that we've been looking at in our projects. And so the first one has to do with carrot, which is one that we're very interested in, again, obviously. And so this is, I'm going to talk about several studies that we've done looking at carrot genotypes and how they interact with beneficial microbes, and specifically with a role in pathogen suppression. So this is part of a large national project that my lab's involved with called CIOA, which is a Carrot Improvement for Organic Agriculture. And so we're working with carrots because they're a very popular and highly nutritious crop, but they also have many production challenges. And I find them particularly interesting as a soil microbiologist because they're a crop that grows underground, so they're very susceptible to things like pathogens and all of these microbes that live in soil. So as part of this project, we're looking at a suite of things from improved weed resistance, nutrient use efficiency, disease resistance, and nutritional quality. But more specifically in my lab, we've been very interested in this idea of genetic differences in interaction with beneficial microbes. And so we've conducted numerous studies over the past 4 or 5 years in looking at these different carrot genotypes from very broad backgrounds, like you can see below. We have Asia, Syria, Turkey, some of this germplasm that we've collected. And so we've been looking at various things. So I'm going to just briefly mention one study where we were looking at genetic differences in responsiveness to endophytes and that pathogen we talked about before, Alternaria dowsii. I'm going to also mention another study we did with endophytic communities that are resistant to pathogenic and susceptible to, or resistant or susceptible to pathogenic nematodes. And then a study we just completed on positive priming of soil organic matter, and I'll talk about why that's important. And then lastly, some feedback effects on disease suppressive or conducive soil. And so we've been working with a wide range, again, of different germplasm from around the world, and we're doing various assays, inoculating with pathogens, or in the case of nutrient scavenging, actually taking crop residues that are labeled with 15 nitrogen so we can track their movement out of these soil organic matter pools and into plants. And so that was a subsequent study another student, Lady Meha, did, where she wanted to know, okay, we're going to take these soils and now we're going to inoculate them with this pathogen, Malloyni-Guinea incognita, that causes all that carrot root galling. And it was really interesting in that she saw these changes in communities really didn't affect the resistant genotype. However, the susceptible one, it was really strongly dramatic. And on the right, as you see, the number of galls. And so both the N fertility, so having a lower rate of fertility, actually there was less nematode galling. And then we had the least and lower nematode galling when the plants were grown after a previously susceptible genotype, indicating that these carrot genotypes were altering the communities around them to create communities that would help them or a next plant to fight the pathogen. So that was really interesting. So next I'm going to briefly mention another genetic study that we're doing along these lines of breeding, and that has to do with tomatoes. So this is another large-scale breeding project we're involved in this case for tomatoes, which are a very important crop, very highly affected by pathogens. So when we talk to farmers, this is the thing they're most concerned about. So we have this whole breeding program nationally about breeding for resistance. And so we're looking for things like disease-suppressive soil, biopesticides, which I'll talk about, with traditional breeding, as well as selection for ISR responsiveness. Again, can we find varieties that are better able to host and respond to these beneficial microbes? So for this, we narrowed it down to one individual microbe, and this is Trichoderma harzianum. You see on the right, it's growing in those petri dishes, producing that kind of green compound. It's very well known for its potential in biocontrol activity, and we wanted to know both how carrot genotypes respond to that, but also how has that changed over the course of domestication. And so the reason that's potentially important is that when we think about our wild crop ancestors, they're growing in environments where they don't have fertilizers and nutrients, and so they're likely to be more reliant on these beneficial microbes to help them to acquire resources or withstand pests. Whereas when we've been selecting for plants for maybe above ground traits primarily, maybe we've forgotten about some of these kind of below ground traits. And so we did these trials with a number of both cultivated tomatoes as well as wild ancestors. And so these are some of the results. The top figure is just the increase in growth of these plant tomato genotypes in response to being inoculated with this beneficial microbe, Trichoderma. And so the ones on the left, those are all the wild kind of plant genotypes, and then we go across in this domestication gradient. And so we saw that, yes, indeed, supporting our hypotheses, the older, wilder genotypes were much better able to benefit from being inoculated with these Trichoderma. So next, the figure below is how then this influenced their ability to withstand Botrytis cinerea, which causes gray mold, a very problematic pathogen in tomato. And you see that the bars are going up and down in this figure. And that means if they're going up, the plant in response to Trichoderma was better able to resist the pathogen. But if they're going down, that means by being inoculated with this beneficial microbe, they were actually more susceptible to the pathogen. So that was really interesting. We didn't really expect that. But what was kind of interesting also, though, is that we also see this kind of steady decline in the wilder genotypes. There was more ability to benefit from the pathogen and help fight disease, whereas in some of our modern genotypes, we saw more negative responses, which indicates while we may be selecting for a lot of good traits, we could be forgetting these good organisms and possibly even causing some negative interactions, and that when we apply these in the field, we could be hurting crops. So again, coming back to the fact that we can think about breeding for beneficial microbe relationships to enhance these more biological control of pathogens. So lastly, and I realize I'm running out of time, so I'll try to get through this quickly, we wanted to look at the survival of these microbial inoculants. So there are a number of biopesticide and biofungicides, tons of them coming on the market now. These can be derived both actually the beneficial microbe themselves or some compound that the beneficial microbe produces, like some of these antibiotic compounds. So there's several different types of these microbial biopesticides or biofungicides, so kind of different modes of action. But again, as I mentioned earlier, the challenge is that while these inoculants can be highly successful in the greenhouse, when we take them out to the field, it's often really variable. So last, I'm going to try to really quickly whip through this study that we just completed that kind of combines all three of these things that we've been talking about. So disease-suppressive soil, these beneficial inoculants, and this idea of breeding. And so we did this study in the context of urban agriculture, which is something we're seeing a lot of growth and interest in here in the Midwest. So lots of benefits from these systems in terms of providing nutritious produce, employment, things like that, but also a lot of challenges. So soils in urban systems can be very highly degraded due to building and things like that, and they can also in some cases be contaminated. So we've been looking at, how can we go into these systems and improve the health of these soils? And one of the best ways to do that would be to focus on using local wastes. And so one great waste product in cities is leaves. And so we specifically looked at a leaf mold compost. And so that is potentially better than other composts because it's going to have lots of carbon, but not as much phosphorus, which can be a challenge of phosphorus enrichment in animal-based compost. And so we wanted to do this leaf mold, which is a more passive kind of composting process, which we thought would increase fungal communities or provide more resources for fungi. So we did a field trial. This is my student Kyle Richardville at the student farm just in West Lafayette, where we wanted to know, does this leaf mold compost improve soil and plant health, but also potentially better support this fungal inoculum, Trichoderma harzianum? And then what about genotypes? So we know genotypes differ in their responsiveness, so how does that play out in the field? And so we did this over 2 years. And this is some of his results. This is just the leaf compost, again, kind of coming back to soil health and how valuable it is. And he saw very dramatic differences. I think if you just look at these pictures on the right, the top pane is no compost tomatoes that are kind of spindly and sad looking, and then the ones below that got these additions of leaf mold compost. So we saw huge benefits in terms of soil quality and saw really pretty dramatic increases in yield, up to 80% over these 2 years, which was a lot more than we expected. So in summary, integrating cover crops, amending soils with leaf mold, anything you can do to enhance the health of the soil is going to alter these microbial communities and reduce susceptibility to pathogens. Crop varieties do differ in their ability to support these beneficial microbiomes, and so there's a real opportunity in breeding programs to start leveraging this and not only selecting for greater yield or disease resistance, but including in that package the interaction with beneficial microbes. And then lastly, we think further efforts to really understand what supports these beneficial microbes in the field that are coming with some of these products will provide yet another layer that farmers can use to ensure that their crops are healthy and to suppress pathogens.